Hello, my name is Larry Milchak, and I am the manager of the Corporate Toxicology and Environmental Science Group with 3M Company in St. Paul, Minnesota. And I'm going to be talking about our efforts at developing alternative methods for inhalation toxicity at 3M. A little bit of background about 3M. 3M has been around since the early 1900s. We're currently selling products in over 200 countries with over 90,000 global employees and annual sales over $30 billion. We are very large from a product portfolio perspective with over 55,000 different products. Those products ranging across multiple different business sectors, including consumer products, industrial chemicals, industrial products, as well as healthcare products and various medical devices. These are some examples of a few of our more well-known innovations and product portfolios, ranging all the way from sandpapers to various tapes, sponges, and obviously the Post-it note product line. In terms of the corporate toxicology group at 3M, we are residing in the Sustainability and Product Stewardship Department. So we are a corporate staff group reporting through the Corporate Research and Development Group. We provide global support to all of the 3M business units. In my group, I've got about 35 individuals. That includes our division support toxicologists, environmental scientists, and those individuals working in our on-campus laboratory in St. Paul, known as the 3M Strategic Toxicology Laboratory, and I'll abbreviate that as STL during my presentation. We are the global centralized resource for toxicology at 3M. We are responsible for coordinating all toxicity testing and human health risk assessments for the entire company. "3M has long been committed to animal use reduction, and I think particularly over the last 10 years, we've made some really significant progress in terms of the replacement of animals in our tox evaluations. I think those can be generically categorized into two major areas. One is the development of our computational toxicology program, and the second is the in vitro method development that we've undertaken within the STL. Obviously, because of the diversity of our product portfolio at 3M, that can add some degree of complexity to incorporating alternative methods, typically because our products oftentimes require global registration requirements. We're going across multiple regulatory categories, and we also have some relatively unique chemistries depending on the specific product portfolio and product line. So I'm going to talk now about those two specific areas. We're going to start first with our computational tox program. And initially, this program began specifically looking at estimates of acute toxicity, primarily for GHS classifications. And this was built around using a collection of software approaches, including QSAR, structural alerts, and read across. We then built a database to store the results of all these assessments, a database that we're still utilizing today. It's greatly improved our ability to go back and retrieve previous assessments, help us with things like read across and so forth. We then expanded our capabilities beyond just acute tox endpoints, started looking at other endpoints of interest to 3M from a tox perspective. And that leads us to the point where we are now, which in essence is deriving and building our own models, typically utilizing historical 3M test data. So specifically with respect to inhalation tox, inhalation toxicity is an area that is of significant concern to us at 3M because it's an area that we want to make sure we are evaluating very thoroughly for some really specific product applications. It also can be relatively challenging from a computational tox approach perspective. If you look at the availability of commercial models, historically, we've only been made aware of one in terms of acute, like LC50 evaluations. That's a software product known as TopCat, which we do have access to at 3M. Now, the exception is within the last, I believe, year or so, I am aware of another software vendor that's launched another acute LC50 model, and we're in the process of evaluating that. But historically, we largely were utilizing the TopCat modeling system for acute LC50 estimation. We went through a project where we looked at our historical test data available to us at 3M, and we ran those chemistries through TopCat and did a comparison with the actual data. And here we weren't trying to arrive at a point where we could actually estimate the specific LC50, but rather just assign that chemical to a GHS acute inhalation tox category. And those categories are shown here on the slide. And to a certain extent, they are relatively broad. What we found was that the model really did not do a great job of estimating the toxicity for our chemistries when compared to the historical LC50 data. We looked specifically at vapors and gases as two separate classes. Specifically with respect to vapors, those predictions from the software were really only accurate 42% of the time, which leads to 58% being incorrect in terms of assigning the applicable GHS category. With respect to gases, it was even worse. They were incorrect in terms of the GHS category assignment, essentially 100% of the time. So clearly that was not necessarily encouraging for us in terms of providing confidence in this model. What we generically found was, with respect to vapors, the model was more commonly predicting a higher LC50 than what the actual data showed, whereas with respect to gases, the model was consistently predicting a lower LC50 than the actual data. So clearly we knew that if we were going to continue to use this as an approach, we had to do something to improve the model. So we started working with a separate software program that actually allowed us to build our own model and incorporate other historical data plus literature search data. And that's still an ongoing project for us. We're still working on improving that model. To this point, what we have found is by including our own historical data, we've been able to improve the confidence in the vapor LC50 estimates by about 34%, with gases improving about 25%. So the improvement is good. It's still not exactly where we would like to be. So we hope to continue work on that, and hopefully those correct estimate percentage will continue to increase. But despite of the lack of concordance we've seen with the LC50 estimates with respect to GHS category, the process has still been very useful for us for other reasons. It has allowed us to identify some key structural features associated with our chemistries, as well as certain physical chemical properties from a computational perspective that appear to have a significant impact on toxicity. So while the LC50 estimates may not be exactly where we would like them to be, the overall process has still been very useful to us because of what else we've been able to learn through the process. Next, I'd like to talk about our internal laboratory and the efforts we've been making there in terms of inhalation toxicity model development. What is the STL at 3M? Essentially, it's an internal on-campus laboratory that is available to 3M business units and research labs. Our goal is to provide quality and cost-effective testing to help businesses develop products and investigate health hazards. Within the laboratory, we've got a significant emphasis on the utilization and development of non-animal alternative methods. We've been doing a lot over the last several years with utilization of three-dimensional tissue models. We also utilize standard 2D cell culture, but we've had a lot of success with three-dimensional models. They allow us to test finished products, formulations that are in development, mixtures, and from that perspective, it's very useful. When we look at inhalation toxicity and model development, we would like to develop an in vitro model to help us screen new chemistries. Ideally, the screening would minimize and hopefully eventually be able to replace in vivo animal testing on some level. We've decided to focus initially on the evaluation of vapors and gases rather than also focusing on aerosols and particles. These are areas of product development that are very important for us at 3M. And that's also an area where we feel that we've got expertise to be able to help build the model and develop the science. We are encouraged with the potential that these three-dimensional tissues that are commercially available may offer for these types of evaluations. And again, given our experience with running these 3D tissues and a lot of other standard assays, like the currently validated OECD methods for dermal irritation, corrosion, ocular irritation, corrosion, for example, we feel comfortable moving ahead with this type of model development at 3M. If you ask where are we trying to go with this, in an ideal sense, it would be nice if we would be able to develop an in vitro model that would correlate well with the classic rat LC50, 4-hour data. But we do recognize that there are limitations to that approach. When we utilize one of these respiratory tissue systems, if it's airway tissue, bronchial tissue, even alveolar tissue, they are representing, in essence, a single target site, a single target system. And acute inhalation toxicity obviously can be manifested through a variety of different systemic organ systems, different mechanisms of action, and so forth. So that's something that we've got to be cognizant of as we try to develop our model. It would be still a great success for us if we can develop a model that will help look at even individual localized effects, irritant properties, and properties that could still have a significant effect on overall respiratory health in vivo. So from exposure system perspective, we purchased a VitroCell system. We've had it up and running for around 2 years now. The system that we purchased is a 12-12 system with a dilution system and a climatic chamber. It's specifically set up right now for us, again, to look at vaporized liquids and gases. And again, that's where we're going to try to focus our initial development efforts. We like this particular system because it does allow us to expose tissues to 3 separate concentrations. We've also incorporated a micro GC or gas chromatograph into the system to allow us to monitor airborne test article concentrations within the exposure apparatus. What have we learned and what have our challenges been specifically with this system? As I mentioned earlier, the tissues we've been working with, which primarily have been available from commercially from MatTech here in the U.S., so we've been looking heavily at epi-airway, epi-alveolar. These tissues are representing a single site and a single target, and we can't lose sight of that fact that when we're dealing with chemistries that may have mechanisms of toxicity outside specifically of the respiratory system, these tissue types in this particular system may not necessarily be robust enough to identify that. Those are areas that we're working through right now. The other thing to think about is the metabolic capability of these tissue types. There can be significant biotransformation efforts within the respiratory system, within the lung tissue itself, with in vivo exposures. and we have to understand the metabolic competency of the tissue type that we're utilizing to identify any weaknesses and specifically any differences that there could be in the metabolic profile between in vivo exposure and the in vitro model that we're choosing to use. The other area that we're spending a lot of effort on right now is dosimetry. In addition to utilizing the vitro cell to conduct vapor exposures, we're also looking at direct liquid applications to the tissue types. And that's important because we have to look at the dosimetry difference between direct liquid application and vapor exposure. What we're seeing initially is that the vapor concentration that we need to achieve to show decreases in tissue viability can be extremely high compared to what we would see from an in vivo animal-based exposure capable of producing toxicity. And that's important as we continue to work with these models because it can present some logistical challenges in conducting these types of exposures in a system like the vitrocell, for example. The other issue is in building these kinds of models, there is no accepted, validated approach at this point compared to dermal irritation or even in vitro dermal sensitization right now. Because of that, we think it is really critical and really important to try to define what is the goal of your experimental approach. Is it to try to build a model that's going to correlate with in vivo LC50 data, or is it to identify specific endpoint-driven toxicity based on local effects given the tissue type that you're working with? And these are questions that we're continuing to dive into on a routine basis as we get more experience with our vitrocell. So in summary, we believe the implementation of these alternative methods has been very successful at 3M. And again, that's been primarily focused on two particular areas, computational tox and development of in vitro methods within our own laboratory. With respect to acute inhalation tox, that is an especially challenging area for method development. On a computational tox perspective, the commercially available models may not necessarily be well represented with your particular class of chemistry that you're most interested in. We've had more success supplementing those models with our own historical data and simultaneously building our own models around historical data. With respect to the in vitro systems, I think if you're going to enter into this type of testing, you do need to understand, just like if you were going to enter into conducting actual animal-based inhalation tox studies, there is a significant amount of time involved with method development. There are a number of factors that you have to consider, primarily including your exposure system, how are you going to be monitoring and measuring exposure concentrations, the dosimetry issues that I talked about, as well as what particular types of endpoints are you going to be looking at with respect to tissue response and tissue viability. For all of those reasons, I think a defined goal really is critical. Try to identify upfront what you really want to accomplish with your experiments and try not to deviate too much from that, or you may end up getting bogged down with some of these other issues. Keep the goal in mind and keep working towards it. From our perspective, we are very comfortable with a lot of the developments that we have made with respect to inhalation tox. We're going to continue to invest heavily in that development because we believe it's good for us to help further and develop the science, and also because we've had success in contributing to product development and helping our business units look at new chemistries with these various approaches. So with that said, thank you very much for your time and your interest.